Hi, and welcome to our second Semel Grand Rounds of the academic year. I'm Steve Martyr, and I'm delighted to introduce our Grand Round speaker, a person who's a friend and colleague to a number of us at UCLA, and also one of the most important figures internationally in schizophrenia research. Uh, Cameron Carter recently came to our neighborhood. He's now the chair and a professor at UC Irvine. That follows a distinguished career at UC Davis, where he had important leadership roles in clinical neuroscience and neuroimaging. Prior to that, Cam was at the University of Pittsburgh. And as you'll be able to tell as soon as he talks, he's originally from Perth, Australia. Cam's stature in the field is based on the fact that he can talk to both neuroscientists and clinicians about psychosis, and he has an understanding of psychosis that's based on his research that includes both clinical neuroscience, neuroimaging, as well as an interest in the clinical high-risk population. In addition to his leadership roles, he's the founding journal editor of Biological Psychiatry, Clinical Neuroscience, and Neuroimaging. He's also a deputy editor at Biological Psychiatry. His career spans a number of decades and includes important work that he did for NIMH in identifying clinical neuroscience indicators that could be used in treatment research. I think that's probably enough for me. So I will introduce Cam Carter, who's going to talk about clinical cognitive neuroscience of psychosis. Thanks, Cam.
Well, thank you, Steve, very much. And, that's a very warm welcome. I'm really happy to be presenting at Grand Rounds. I think this might be the third time over the years that I've presented. As you mentioned, I'm now in the neighborhood in Orange County. If you look out my window, that is Disneyland that you can see in the distance. And also very much enjoying being in Southern California, building my lab here, and working with a department that's in an exciting kind of growth phase. So again, thank you very much for your kind introduction. I'm going to share my screen and begin my talk. So I have no disclosures. And today, my talk is going to be couched in the context of schizophrenia as a neurodevelopmental disorder, and a key premise for my research approach. I'll talk briefly about a circuitry level account of the cognitive and disorganization symptoms of schizophrenia, and of which I've spent, a considerable part of my career investigating. And then I'll talk about three different areas that I would consider to be extensions of this more cognitive neuroscience-oriented research, both into important clinical questions, such as how could we potentially use neuroimaging to help us guide treatment selection in schizophrenia, work that's more preclinical and translational. It's focused on links between neural and cognitive features of schizophrenia and non-genetic risk factors associated with the illness. The focus there will be on maternal exposures, and this has been the focus for almost 10 years of the Conti Center that we continue to have at UC Davis, and of which I continue to be involved with as a multi-PI. And then finally, I'll talk about new work just very recently published that explores links between clinical and neural features of schizophrenia and the potential genetic origins of the illness. Just a very quick nod to the schizophrenia syndrome. It's common. It has a slight male preponderance. It affects people in adolescence or early adulthood. There are core symptoms that we can deconstruct that are dimensional in nature, positive, negative, and disorganization symptoms that all contribute to the suffering and disability that's associated with this illness. Most of the time, it's associated with impaired social and occupational functioning, and our treatments don't typically produce full remission, and treatment resistance, treatment non-responsiveness is all too common, even when we bring out the big guns and provide multimodal interventions for our patients, and that's very costly to the nation. So the notion that schizophrenia is a neurodevelopmental disorder is something that was actually kind of a novel idea early in my career, because I was educated under more of a Kraepelinian view that schizophrenia was this early onset kind of dementing illness. And I was intrigued as these important studies appeared really at the very beginning of my career. So the first one was work by Elaine Walker, who did some very creative work looking at home movies of children's birthday parties, and had trained raters basically identify the kids that would be most likely to be destined to develop schizophrenia in adolescence and young adulthood, and to distinguish them from the other attendees of the party that were not. And they were well better the chance of doing that. And the raters were responding to features of the home movies of birthday parties, such as reduced affective responsiveness, awkward social interactions, and motor awkwardness. And this work has been replicated and was one of the early, I think, at least high impact clinical studies that really, made the point that there's an echo, a developmental echo that's there prior to the onset of the illness, at least at the population level. And there's been lots of other studies, many studies. I just mentioned a couple. One is the Dunedin study, 1,000 individuals recruited at birth, followed throughout childhood and now on into adulthood. And important work that Mary Cannon and Caspian Moffat did with this cohort. This first very highly impactful paper where they showed that the young people that were later destined to develop schizophrenia actually showed a wide range of behavioral differences compared to those who did not, or who developed mood disorders, including motor awkwardness, behavioral symptoms such as internalizing and externalizing symptoms, and cognitive functioning measured using standardized methods, on an ongoing longitudinal basis. So again, and then one of my favorite studies, and I actually can't see who is in the audience in my view right now, but your own Bob Bilder did this really interesting follow-back study in the early part of the century, where they took a cohort of recent onset patients and demographically matched controls, and they tracked them back and looked at their performance on standardized tests in the educational setting, starting in the first grade. And what's quite striking is, again, as in the Dunedin study, early in life, in this case in the first grade, people who later went on to develop first episode patients were already at about 1 year behind in terms of school achievement. So this, and of course, many, many other studies have made the case that at the population level, there are features, behavioral features, cognitive features that indicate that those that are at very high risk for developing the illness, or actually do go on to develop the illness, already show developmental deviation from those who do not. So why study cognition in schizophrenia? This is a slide that I've showed in various forms for most of my career. It's not really a curious question now, but it was a bit more curious back in the 1990s when I was getting started. So impairment in cognition is present at the illness onset. It's also present in risk states. And as I've already mentioned, it's seen in cohort studies of people who go on to develop the illness. It's consistently and uniquely associated with functioning and functional outcome in schizophrenia. Current treatments have very limited efficacy. We haven't really got, a standard approved treatment for impaired cognition. There are promising approaches using cognitive training and neuromodulation, but these haven't generally penetrated the clinic. I know at Keith's clinic at UCLA, this is offered. I think that's a real benefit to the patients there. The tools or constructs of cognitive neuroscience, the theory, the tasks that we have, make understanding changes in brain circuitry tractable. It's feasible to conceptualize and measure cognitive functioning in people who have clinical disorders, and to combine that with methods for recording brain activity during cognitive performance. And this has opened up a whole world of understanding the altered neural circuitry that's associated with different aspects of serious mental illness such as schizophrenia. And it really opens up the potential for developing circuit-level biomarkers and targets for intervention. In fact, I think I mentioned to one of my colleagues a few weeks ago that in some ways, neuromodulation itself is kind of applied clinical cognitive neuroscience. And this is the circuit psychiatry that Josh Gordon has talked a lot about. And I'll touch on some approaches that we have tried to do that are ongoing in our lab, both at Davis and now here at UC Irvine, to try to leverage or to use cognitive neuroscience-based measures as bridges to better understand the basis of clinical symptoms, but also as potential biomarkers for predicting treatment response and as bridges to better understanding the cellular and molecular basis of the illness. And that's kind of what I say there. So just very quickly, the notion of cognitive control, which has been the aspect of cognitive functioning that I have focused on for much of my career. Basically, cognitive control is needed when habitual responding won't do, especially if you're in a situation where there's a strong tendency, a prepotent tendency to respond habitually, and you have to use a representation of a task or a goal to overcome that prepotent response to guide behavior. Good example is if you've been to the UK or other places of the world where they drive on the left side, you come out of the tube. If you don't remember to look to the right before you step into the traffic, you could really come to a sticky end. And cognitive control is usually measured using cognitive tasks from cognitive psychology, the classic Stroop task that goes way back into the 1940s, where you have to overcome the prepotent tendency to read this word and say the word red when you've been instructed to name its color, blue. So when people with schizophrenia get a cue that tells them that they're going to need to use cognitive control, and actually healthy controls doing this task, they'll activate a frontal parietal premotor network. And when you compare healthy controls to schizophrenia, as John Yoon did back also, in the early part of the century, during fMRI, doing a task that engages cognitive control, in this case, it's the AXCPT task, people with schizophrenia fail to activate this circuit, and they also have reduced connectivity within this circuit with the dorsolateral prefrontal cortex. And these reductions in activation and these changes in connectivity within this network are strongly associated with aspects of the behavior of patients, including reduced task performance, so failure to activate, poor performance, good activation, better performance, behavioral disorganization as measured, across SENS and SEPS items that are associated with this construct of disorganization, and also less functioning, measured by the GAF. And as I mentioned, we've spent a fair bit of time over the decades drilling down on this aspect of psychotic illness. And we know from our studies and many others that it's impaired at or prior to the first episode, seen in clinical high-risk youth. It's related not just to the diagnosis of schizophrenia, but it's also seen in bipolar illness. The level of impairment, both behaviorally and in terms of frontal parietal function, tends to be intermediate between people with schizophrenia and people and healthy controls, so consistent with a transdiagnostic dimensional aspect of these two syndromes. It doesn't remit or progress during the early course of the illness. One of the mysteries, I think, of schizophrenia, and something that we need to understand better, because patients clearly do lose functioning over the first several years of the illness. But in our hands, using longitudinal behavioral and neuroimaging assessments, we've seen no evidence of either progression or remission of this aspect of the illness. And in fact, what we do see is in younger patients, they continue to develop, they continue to improve cognitive control performance and brain activity, suggesting that they still have potential for brain plasticity and cognitive and neural development, despite the fact that they've developed a psychotic illness. And they increase more or less at the same rate that healthy controls, but those are parallel lines. They never catch up in the studies that we've done. We also know that poor frontal parietal based cognitive control is associated with poor treatment response, and I'll show you some data about this shortly. And so our interpretation, our inference, if you will, related to this aspect of the illness is that it's a potential biomarker that reflects processes that are related to the pathophysiology of the illness. And since they're present earlier, prior to the onset of the psychotic symptoms, they likely have a developmental basis. And it's associated with both psychosis and psychosis risk. And so this first body of work, which actually is still very much a work in progress, is related to this aspect of schizophrenia, the heterogeneity of the illness. Here are three publicly known people: Nathaniel Ayres, John Nash, and Ellen Sachs, who, according to DSM-5 and their own recognition, one way or another, they have schizophrenia, and yet their clinical course and their response to treatment is each unique to them and quite different from one another. And one of the things that we were looking at a number of years ago in our first episode clinic at UC Davis, the EDAM clinic, was, we were big believers in coordinated specialty care. We're committed to that approach. We do see better than expected outcomes in our patients. But when we looked and we used a criterion of a 20% reduction in BPRS symptoms across the first year of CSC treatment, we were kind of disappointed to see that about 40% of our patients didn't show that pattern. And so I've become very interested in trying to understand how we might determine, using the tools that we have, clinical assessments as well as neuroimaging, who is more likely to not do well with our standard package of interventions and might need unique, personally targeted interventions in order to get a good response, because 40% is, given the intensity of care that we provide in CSC, is very disappointing. The use of neuroimaging for diagnosis, of course, is controversial in psychiatry. And a number of years ago, I was involved in this project. Actually, it played out over 3 or 4 years, where we did a very extensive review of neuroimaging, asking the question, are we ready yet to use neuroimaging to inform diagnosis of treatment in clinical care? And the answer was a decided no at that point. Jason Smusty, who's a junior faculty member, a KORD, who's working at UC Davis, and I continue to work with him as a mentor, did this very interesting analysis, though, where he combined a couple of cohorts data that we had collected over a period of probably a decade at UC Davis. And he asked the question, is there a relationship between frontal parietal activity during the AXCPT and whether or not people are in responders or non-responders over 1 year of coordinated specialty care treatment? And what he found was, so this is the dimensional analysis, if you will. This is a correlation between the improvement in total BPRS score and the standardized score for frontal parietal activation. And in that study, he saw a pretty robust relationship. And that was true in this analysis, or more categorical analysis, where we divided people up into responders and non-responders based on the 20% reduction in BPRS, and did logistic regression, which was highly significant as well. So if you have a good prefrontal cortex, when you are enrolled in this study, which is at the beginning of your year of coordinated specialty care, you have a much better chance of being in that responder group and having a significant improvement in your BPRS score over the year. And of course, there are many reasons why this could be so. It wasn't related to medications, though. So a second potential pathophysiological measure that we have also begun to focus on, with the hope of developing biomarkers that could help predict treatment response, is dopamine, where we know in schizophrenia that in general, people with schizophrenia tend to make and release more dopamine in the basal ganglia than healthy control subjects, and this tends to be related to the level of psychotic symptoms they have. And there were already early on some data suggesting that people who made and released a lot of dopamine actually tended to respond better to dopamine-blocking drugs than those that did not. And there's a literature actually largely out of the King's College in the UK, this particular study, and actually several related studies that tend to converge on the observation that people who are responders to standard treatments tend to have the increased dopamine synthesis capacity measured using PET, and that non-responders tend not to. And this is another way of looking at that. And so we've become very interested in exploring dopaminergic function as a potential biomarker, with the notion that there may be a subset of patients that don't have excessive dopamine and might be less likely to respond to our standard treatments. And we're using neuromelanin-sensitive MRI, which many of you might be familiar with. Neuromelanin itself is a pigment, a byproduct of catecholamine metabolism. It accumulates over time in the substantia nigra and the locus coeruleus. It's a proxy for dopamine function in the SN, and norepinephrine, the catecholamines in the LC, and has been very standardly, very well, I think, validated as a proxy measure, particularly by Cliff Cassidy and Guillermo Horga. It's a relatively straightforward to acquire. It's a gradient echo, a magnetization transfer prepared, sort of high resolution slab that you get in the midbrain. And then there are various manual and automated ways of measuring the intensity of the neuromelanin measure. In our hands, in a preliminary analysis, we do see a tendency for patients with schizophrenia to have a higher neuromelanin. Obviously, lots of overlap between the groups. There's meta-analysis. There's a recent meta-analysis a couple of years ago that across subjects suggested that's effect size of about 0.35, 0.4 increase. It's across the published studies. And again, as part of this recent preliminary analysis that we've conducted, we do see that neuromelanin is associated with the reality distortion syndrome score, which is primarily hallucinations and delusions, but much less so disorganization, and not at all for poverty syndrome negative symptoms. So in our own relatively early course of illness cohort, we have seen that neuromelanin levels actually are associated with the intensity of psychotic symptoms, an important clinical feature. And then we do have preliminary data that I'm showing you here that treatment responders also tend to have higher neuromelanin levels than non-responders. And the third measure that I'm going to quickly talk about, that's part of this larger study that I'm coming to describe to you, is extracellular free water. It's a diffusion-weighted imaging method. We use multi-shell DWI, and we do an analysis that essentially segments water that's in the tissue compartment from water that's in the extracellular compartment. The free water measure is increased in animal models of inflammatory activity in the brain, and also in human neuroinflammatory disorders such as MS and Alzheimer's disease. And there are a number of studies, starting with the initial study by Pasternak and colleagues, that have shown that it's increased in schizophrenia. This is work that we published in Molecular Psychiatry a few years ago, I think 2019, where we showed elevated free water in the cortical gray matter in an early course of illness population. And we also showed in that study that the level of elevated free water in the cortex in our population with schizophrenia was inversely correlated with glutathione, which measured using magnetic resonance spectroscopy. And glutathione is a free radical buffer, and its levels would be expected to be decreased in the presence of inflammation. So this is some additional evidence that, in fact, the measurement of free water in people with schizophrenia may be related to something that is neuroinflammatory. And then more recently, this is work that's not published, I think it's under review right now. We've shown that treatment response is also associated with free water levels, such that patients with higher free water tend to be more likely to be responders than patients with lower free water, suggesting perhaps that a high level of whatever neuroimmune perturbation is going on that might be conceptualized as inflammatory. And we can talk about that word. It's not a good word, I think, to describe what we think might be happening in schizophrenia, but it's probably the closest to a good word. That might be a better kind of schizophrenia associated with better outcomes in this early psychosis group that's participating in coordinated specialty care. And so that takes us to the project, which we're about probably 2/3, 3/4 of the way through. Any of you who do clinical research probably had some challenges during the pandemic, but we sort of rectified our challenges, and we're recruiting patients both at Davis and UCI right now into this study. And we're looking at these 3 putative biomarkers: a developmental prefrontal cortical cognitive control circuitry measure, excessive dopamine production reflected by neuromelanin imaging, and neuroimmune perturbation, neuroinflammation measured using diffusion-weighted imaging. And we're seeking to replicate the findings that I just presented to you. That's sort of the first part of that. And then I think the second part is to, in collaboration with my colleague Ian Davidson in the computer science department at UC Davis, develop a learner, a neural network, a form of deep learning artificial intelligence that can use these features and potentially other features, either singly or in combination, to do a better job and maybe get us to where we would like to be with 80% positive and negative predictive value, to figure out when people come into treatment, whether that's early psychosis treatment, which is my focus, or maybe later in the illness, whether someone is likely to respond to our standard therapies, or whether they need some more creative approach. Perhaps that could be just early transition to clozapine. We are getting closer to having non-dopaminergic antipsychotics, which is exciting. But hopefully it can inform those clinical decisions, and we can get better than 60% treatment response rates in our care of young people with psychosis. All right, so that is those three areas, and that study that I described is our effort to try to take neuroimaging, which is a pretty mature science in clinical neuroscience. We've got a 30-year track record or more using some of these methods, and to bring it into the precision medicine realm. And of course, it's very challenging, and I'm happy to discuss with you the limitations and the promise that this particular approach would have. The second area of work that I'm going to describe to you is work that we've done as part of our Conti Center, and it's related to non-genetic risk factors for psychosis. It's more translational in the nature, but we're very much still informed by our cognitive neuroscience perspective and the neuroimaging tools that we use in patients. Its focus is on neuroimmune mechanisms, and particularly maternal exposures as an etiology for psychosis, and as a model for psychosis, Yeah, and our goal is to get insight into how something like having the flu during the early phases of pregnancy could cause changes in brain development that would render the brain more vulnerable to developing serious mental illness during adolescence. So the rationale for focusing on neuroimmune mechanisms include, the widely reported exposure to infection during pregnancy that's associated with increased risk of offspring psychotic disorders. The fact that this increased risk isn't related to a specific infection, but it very much appears to be related to the mother's immune response, the strong association with immune genes in schizophrenia, and the neurobiology of these molecules and their role in brain development and function. Immune molecules play a very wide-ranging role beyond what we would conceptualize as traditionally immune responses during normal brain development, as well as in maintaining healthy synaptic function. So these molecules are very important. Anything that would change them has the potential to really change the function of the developing and adult brain. There are rodent models that recapitulate some developmental phenotypes related to maternal exposures. And there's some evidence, it's not very strong in my opinion, but that anti-inflammatory therapeutics might be helpful in some patients with schizophrenia. In this Conti Center, you are probably all familiar with the P50 model. There are multiple projects. We've all been focused during the first 5 years, and now we're into our renewal phase on this hypothesis that alterations in immune molecules during brain development lead to changes in connectivity, and that underlies the emergence of symptoms and other serious mental disorders. And I'm just going to focus on these two projects, Project 3 and 4, because these studies involve longitudinal neuroimaging using many of the same tools that we use in our patients, as well as very detailed phenotyping and cognitive assessments in a cohort of macaque monkeys that have undergone maternal immune activation. And this work is led by Melissa Baumann, and David Emerald also played a role in the early phases of our center. Basically, the model itself involves administering poly-IC-IC, which is a synthetic RNA, late in the first trimester. That timing was based on fairly extensive pilot work that Dr. Baumann conducted. And the dams, the mothers, get sick when you do that. They get a fever, they stop eating, they show other kinds of sickness behaviors, and they have a very robust cytokine response during that period. And all of that is measured in our animals. But then they go on and they deliver normally, and the infants then are housed in a fairly enriched environment, and they undergo very careful observation and very deep phenotyping. This includes behavioral observations, longitudinal cognitive assessments starting quite early in life, and neuroimaging, which is longitudinal and starts at 6 months and then is repeated at 12 months and then annually until adolescence. And just to, in a nutshell, talk about a very large body of work, which continues to be data analysis is ongoing, and papers are in preparation. But basically, the offspring who receive maternal immune activation are clearly atypical, but not until they get to be about age two or so. In terms of general, maternal interactions, social interactions, etc., these animals are not really distinguishable from their cage cohorts compared to those that are sham treated. And around about age 2, there begin to be seen some subtle cognitive impairments and social deficits. The cognitive impairments are largely, I would say, complex. Probably, one would understand them as involving significant levels of cognitive control, and they're primarily reversal and set shifting findings that are measured behaviorally. Now, in contrast to the cognitive and social deficits, the brains of these young monkeys actually are atypical, starting from the beginning that we start scanning them, so at 6 months. And the findings are selective reductions in frontal cortical volumes, both prefrontal and more broadly frontal cortex, and increased free water measured using diffusion-weighted imaging in the medial frontal cortex that's present. And all of these are present as early as 6 months. And in the case of the free water finding, very interestingly, it's strongly associated with the level of the maternal IL-6 response that we measured when the monkey was undergoing maternal immune activation, providing some more evidence that the free water measure is reflecting neuroimmune or neuroinflammatory mechanisms. So early on, we see these changes in brain structure measured using similar methods that we use in our patients. We also see evidence of dysregulated dopamine, increased dopamine. This is present at age 2, and we see this during FMT PET imaging. And then later, prior to adolescence, we see some reductions in white matter changes. So these other things that I talked about, they're observed and they're fixed and they persist all the way through adolescence. The reductions in frontal white matter aren't present prior to age 3, so they are observed at age 3 and 4, suggesting there's some progressive developmental changes happening associated with these more gross measures. Preliminary cellular molecular analysis is very interesting. Work from Cindy Schumann's lab show changes in spine morphology in the selected to the prefrontal cortex, actually, that are present in the adolescent monkeys. And Mike Gandel, Dan Geschwin, who are doing the transcriptomic work, have shown changes in gene expression that are largely more, synaptic and developmental than inflammatory. However, I think some very recent work that's happening in Dan's lab using cell-specific transcriptomic does suggest that at least in some circuits, we're seeing evidence of neuroinflammatory processes underway as well. So this is interesting, exciting work that's very much, ongoing. What I think this overall approach tells us, and these early findings from our Conti Center tell us, is that there's pretty strong value in this model. It appears to have construct validity for understanding the neurodevelopmental impact of maternal immune activation and potentially maternal infections and their potential relevance to developmental disorders in general and schizophrenia in particular. Then the very last thing that I will present here, hopefully leave time for questions, is a project that we just wrapped up and published, a collaboration between myself and Tyler Lesh and other members of the lab, and Alex Nord, who is a neurogeneticist at the Center for Neuroscience at UC Davis, and Tracy Warren. He's a grad student who just graduated, and is, I think, at Yale doing a postdoc, and Pak Sham at University of Hong Kong, and his student at the time, Justin Tubbs. So, and this work basically had as its goal to test relationships of diagnoses, clinical diagnoses, as well as clinical measures, more dimensional clinical measures, and cognitive and imaging phenotypes to the polygene score for schizophrenia. And an important goal from the beginning here was to also look more closely at pathway-specific polygene scores that were related to major psychosis-associated synapse types in the brain. So in addition to the overall polygene score, we computed a dopamine polygene score, a GABA polygene score, a glutamate polygene score, and a serotonin polygene score. This is some information about the cohort. So it was around about 120 people with schizophrenia, 39 with schizoaffective disorder, 48 with bipolar disorder, and 115 healthy controls. And the measures that we had were a measure of treatment response, the 12-month BPRS change that we talked about, 20% reduction, functioning measures, cognitive control, both behavioral and frontal parietal network activity measures, and then clinical measures of disorganization, poverty syndrome or negative symptoms, positive symptoms / reality distortion, and mania. And this is some details that I'm sure will be important to many of you, and we can come back to them if you like, but these are the general methods, the genetic methods. And here's the findings. So one thing we saw, as a non-geneticist, I loved this first result. It might seem silly, but it makes me feel like what we're doing, with our diagnosis might also be right in the clinic. So we saw a strong relationship between schizophrenia polygene score and the presence of psychosis. We also saw that this was elevated in schizophrenia and schizoaffective disorder. Not bipolar disorder, although, it is a relatively small n. Interestingly, we did the same thing with the bipolar disorder polygene score, and we actually saw that it was related to that bipolar illness was related to the bipolar polygene score, and the other two, schizophrenia spectrum diagnosis, was not. But with the overall schizophrenia polygene score, we actually didn't see any robust relationships with any of our phenotypic measures. Let's go. Treatment response, functional measures, mania, psychotic symptoms, and cognitive control measures. Really no significant findings there. So, that's maybe surprising to some because there are some studies that show relationships between clinical features and polygene scores that are published. There's also some studies showing cortical thickness relationships with polygene scores, but there also are quite a few studies that don't show relationships between polygene scores and cognitive phenotypes in schizophrenia. So, I know that many people doing this work at UCLA, and I hope to have some good questions and comments about this. We went on and we looked at the pathway-specific polygene risk, however. And what we saw was that for the glutamate polygene score, it was elevated in people with schizophrenia, not in the other diagnoses. For the GABA polygene score, it was elevated in schizophrenia, but not in the other diagnoses. And for the dopamine polygene score and the serotonin polygene score, it was not associated with any of the diagnoses. However, we did find that the dopamine polygene score was associated with poorer global functioning. So people with a higher polygene score had lower functioning, which was interesting. And we also saw a finding that I really liked when we saw it was that, consistent with the imaging data and other treatment data, a high polygene score was associated with a higher likelihood of being a treatment responder in our first episode cohort. We also saw that glutamatergic risk was associated with our cognitive control measures, not dopamine, not GABA, and not serotonin. So both for D prime context, which is behavior in the scanner, and for DLPFC and superior parietal cortex, all of those measures were associated with a higher glutamate polygene score. Okay, to just summarize those studies, schizophrenia polygene score is associated with schizophrenia and schizoaffective disorder. Glutamate, GABA polygene scores were associated with schizophrenia, and dopamine was associated with treatment response. So conclusions and future directions. So this presentation was sort of organized around the premise that cognitive neuroscience is a basic science, and the approaches applied to clinical research can provide traction, addressing major problems facing the field of schizophrenia research, including the need for personalized approaches to treatment, as well as for implementation in translational research that seeks to understand the etiological and pathophysiological mechanisms that underlie the development of this common and disabling illness. And also that future directions for us as a team will include expanding cohorts for deep phenotyping and precision therapeutics. So I'm hoping that we can have a conversation amongst the schizophrenia researchers in Southern California. Steve has already started bringing us together for scientific interactions about how to expand our ability to get deep phenotypic coverage in larger and more diverse cohorts of patients, and try to test this idea out, and perhaps do a better job of taking advantage of all we've learned through neuroimaging and cognitive neuroscience over the past few decades. And then there's many people to thank, and I can't possibly include them all, but this is the team from the McConty Center And you can see your own Dan Gishwin there, and Mike Gandel has moved back east now, but he was a key contributor early on. And the initial leaders of the Conti team, Kimberly McAllister, David Emerald, and Melissa Bauman, and a very important people from my group, Tyler Lesh, who is also a UCLA alum, and Jason Smusny. So that's my talk, and I'll stop sharing, and I'm hoping that I've saved some time for some questions.
Thanks so much for a very interesting talk, Cam. Please, if people want to ask questions, please put them in the chat. I'm sorry, but add them to the Q&A. Cam, I'll start off with a question. If you go sort of outside the data and look at it from above, it almost seemed to me that you were putting together a theory of a particular subgroup of people with schizophrenia who had deficits in cognitive control, that was associated with impaired circuits, which in turn was the result of sort of a inflammatory process that occurred during brain development. Am I putting too much on this, or is this where you were going, and that in itself would be associated with a response to a dopamine, a D2 antagonist?
So, not quite, Steve. I mean, I do think that the Conti Center work is focused on risk states, and I do think that the frontal parietal developmental dysfunction is associated with risk states in general, with risk for the illness. But as far as people with established schizophrenia, I don't know that all those things neatly fit together. You know, it could be that there are three pathophysiological mechanisms amongst many potential pathophysiological mechanisms that we can identify and measure using our clinical neuroscience tools. And they may or may not be related to one another. So we actually haven't tested that out. We are getting to the point now where we have a reasonable sample of people at their baseline that we can look for relationships between the dopamine measures and the free water measures and the PFC physiology, but we don't know that. And so I think yes to the first part about the developmental process. And Dave Lewis and Jason and myself have a paper in Neuropsychopharm Reviews, I think from a couple of years ago, that kind of lays that out and includes a lot of Dave's work on prefrontal development. Not so much the focus on neuroimmune mechanisms, but I agree with you about that conceptualization. And then I think, you know, but I don't think everyone who has, you know, we know from the clinical high risk data that as a group, clinical high risk patients tend to have as much of a change in PFC, frontal parietal activity, as people with established illness, and yet most of those don't go on to develop psychosis. So there's more complexity there. And then I think once we get to psychosis, there may be multiple mechanisms. I think that's highly likely, and they may or may not be interrelated. I mean, that's for the future.
And do you see cognitive control as being how do you, I mean, in your mind, how do you sort of relate a loss of cognitive control with reality distortions? Or is that going too far?
No, I would say it must be quite complex, because if you just have cold cognitive control tasks, they typically pretty reliably correlate with disorganization, but sometimes with negative symptoms, especially if the task has some kind of emotion or reward component to it, but not ever that I've ever seen with positive symptoms. So so you'd say, well, it has nothing to do with it. And yet, you know, there's very interesting work going on now, you know, looking at, you know, perceptual decision making and other mechanisms that might be altered in schizophrenia that are related to hallucinations and delusions. And in those studies, some of this same circuitry, and as well as sort of additional different circuits in the brain, are implicated in that altered decision making. So, and the use of prior experience and prior probability and those sorts of things to make decisions about whether they're hearing a voice or noise, or whether that person is following them or not. So it might be that it'll all connect at some point, but just at its simplest level, cold cognitive controls pretty much aren't related to psychosis.
Okay.
And in our first episode cohort, and it's relatively modest, we didn't see that symptom severity was a strong predictor of whether people got better or not at a year.
No, I would agree with that. As a clinician, symptom severity is really not related to clinical populations to response to treatment. I agree. There's a question from Diana Lee Luxembourg, and I'm not sure I understand it. I hope that you do. It's two questions. Were there weight differences in the primate studies, and what were the social deficits?
Yeah. So the Mia monkeys were a bit smaller, not observably smaller, but they had less body mass, and all of that was included in all of our analyses. They also had slightly smaller brains, and we did global correction for that in all of our neuroimaging analyses. So I hope that's a clear answer to the first part. Yeah. So the social. So Melissa Bauman collected the social measurements, social behavior measurements, and she's still analyzing them. But one of the things I think that is maybe one of the earliest recognizable behavioral changes is changes in behavior in relationship to an unfamiliar animal that's introduced. And the Mia monkeys tended to show, surprisingly, more approach behavior than the untreated. There are, a number of, there are eye tracking data. There's lots of other data related to social functioning, and it's just not ready yet to talk about. Sorry, but I will say, Steve, that since I'm delighted at the attendance, but I don't know who's here. Let me encourage you to shoot me an email or give me a ring if you want to, talk about these things in more detail, if you're excited about this work. So let's go ahead with the question, Steve. Thanks.
Yeah, and the formula, you were referring to the Oliver Howe's group, I think, and in talking about the treatment responsiveness as being related to having a more dopaminergic illness compared to those. Where do you think that sits now? Is that overly simplistic? Does it fit at all with your? How do you fit it with your data, with your findings?
Sure. Well, I think there's sort of two pieces to Dr. House's kind of model. One is that there's reduced, that there's, in the treatment responders, they're the ones that are making and releasing too much dopamine. And then in the treatment non-responders, he has some magnetic resonance spectroscopy data suggesting that maybe they have elevated glutamate. I think the evidence is more consistent for the PET stuff. Obviously, most of the published studies, include his group. You want to see more replications outside, and the story with dopamine and PET has gotten more complicated because there's some more recent studies that show populations of patients that aren't showing increased dopamine. So understanding those differences, across different cohorts is, I think, very important for the field. But I do think that it's a valuable starting point. That perspective is a valuable starting point, and it helps us to frame hypotheses and then to test it in different samples and using different methods.
Okay, well then, let me close and to thanks Cam for a very interesting and provocative talk. And now that you're in the neighborhood, we hope to have a lot more conversations with you.
That sounds
Thank you.
wonderful, Steve. Thank you. Thank you all for attending.
Sure. Bye.